Bismillahirrahmanirrahim. Assalamu alaikum. This is Ms. Shumala Kadir, conducting your course 317. Today we have our topic Hexose Monophosphate Pathway. First of all, see the other name of this pathway, why it is called by other names too. Hexose Monophosphate Pathway is basically the alternative pathway for glucose oxidation. Most of the glucose, or somewhat 90% of glucose, is oxidized by glycolysis. But HMP pathway is also going on for oxidation of glucose to a certain extent. This pathway is also called glucose monophosphate shunt or HMP shunt because a glycolysis process is shunted and glucose 6-phosphate entered in the hexose monophosphate pathway or HMP pathway or shunt. It's also called pentose phosphate pathway because a number of pentose sugars are formed in this pathway, xylulose, ribulose, and which also form other pentose like arabinose, and their functional group changes or they are epimerized with their other form of pentoses like ribulose converted to ribose, and so the keto group, the keto sugar converted to aldo sugar, also the ribulose converted to xylulose, that we will see later. And the intermediate of this cycle also participate in a number of pathways for the synthesis of different compounds. And that is why it is also called pentose cycle. The first stable compound which is formed in this pathway is phosphogluconate. That is why it is also called phosphogluconate pathway. Let's see the cycle. Glucose 6-phosphate. Here, the 3 molecules of glucose 6-phosphate enter the HMP shunt or HMP pathway, and that is glucose 6-phosphate, which is also the part of glycolysis, enter the HMP pathway where the 3 molecules of glucose 6-phosphate converted to 6-phosphogluconolactone. With the presence of enzyme glucose 6-phosphate dehydrogenase that require NADP. NADP act as hydrogen acceptor. So NADPH, H plus are formed as 3 glucose 6-phosphate molecules enter. So 3 NADPH, H plus are formed, and it is occur in the presence of divalent ion as a cofactor, that is magnesium ion, manganese, or calcium ion. Now what happened? This is basically unstable molecule, that is 6-phosphogluconolactone. So it is converted to 6-phosphogluconate by addition of water molecule. So required the hydrolase, that is gluconolactone hydrolase. So in presence of gluconolactone hydrolase, one water molecule is added and the stable molecule, that is 6-phosphogluconate is formed. Again, it is required cofactor magnesium or calcium ion. Another oxidation occurs when 6-phosphogluconate is converted to 3-keto-6-phosphogluconate, which is later on converted to ribulose 5-phosphate. The 6-phosphogluconate is converted to 3-keto-6-phosphogluconate in presence of enzyme phosphogluconate dehydrogenase. And so here 3 more NADPH H ion are formed. So total 6 NADPH and H ion are formed. Now decarboxylation occurred and this 3-keto-6-phosphogluconate is converted to ribulose 5-phosphate, a pentose sugar. From here, the non-oxidative stage of HMP shunt is started. Previous of this, the stage was oxidative. Now ribulose 5-phosphate is converted to any diol form. First, which is intermediate and later converted to ribose 5-phosphate. This anidiol is basically the form where two hydroxyl group is attached to the carbon atom, which are attached with each other with double bond. Now this ribose 5-phosphate has two options. Phosphorus, in the presence of phosphotransferase, it can be converted to ribose 1-phosphate. Phosphate group is transferred to one carbon. Or it can be phosphorylated. Another phosphate group is attached in presence of magnesium ion. It then forms ribose 1,5 biphosphate because two phosphate group are attached. As 3 molecules of glucose 6-phosphate entered, so here the ribulose 5-phosphate, which is formed, will lead to formation of 2 molecules of ribulose 5-phosphate and other molecule or other pentose sugar, that is ribose 5-phosphate and allulose sugar. So 3 molecules of pentose sugars are formed, 2 xylulose, 1 ribose. Now this xylulose 5-phosphate and ribose 5-phosphate can form pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. What happened there? In presence of enzyme transketolase, the keto or the keto group, the 2-carbon unit transferred to allulose sugar, that is ribose 5-phosphate, which form pseudoheptulose 7-phosphate, and the remaining 3-carbon unit form glyceraldehyde 3-phosphate. And this reaction requires thiamine pyrophosphate as coenzyme and magnesium ion. As it is multicyclic process, this pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate take part in another reaction where trans-allulase enzyme work, and this trans-allulase enzyme causes allole cleavage where 3-carbon unit form pseudoheptulose 7-phosphate dihydroxyketone transfer to glyceraldehyde 3-phosphate. So they form 6-carbon containing sugar fructose 6-phosphate, and the remaining 4-carbon unit of pseudoheptulose 7-phosphate thus form ethose 4-phosphate. Another transketolation occurs where the xylose 5-phosphate, the remaining xylose 5-phosphate, because 2 xylose 5-phosphate were formed. So here again the xylose 5-phosphate react with this ethose 4-phosphate, and here again the keto group from xylose 5-phosphate transfer to ethose 4-phosphate and results fructose 6-phosphate, and the remaining one from the remaining 3-carbon unit is now glyceraldehyde 3-phosphate. And again transketolation require here thiamine pyrophosphate and magnesium ion. So total 2 fructose 6-phosphate are formed with glyceraldehyde 3-phosphate formation, that is 1 molecule of glyceraldehyde 3-phosphate. For your better understanding, let's see once again the reaction with their structures to make you more clear. And the 3 glucose 6-phosphate molecules enter the pathway, which then converted to 6-phosphogluconolactone in the presence of enzyme glucose 6-phosphate dehydrogenase, which remove the hydrogen ion, and that is accepted by NADP and form NADPH H+, and this 6-phosphogluconolactone is unstable, so converted to 6-phosphogluconate. Again, this 6-phosphogluconate first form 3-keto 6-phosphogluconate, which then form ribulose 5-phosphate. Here 3 ribulose 5-phosphate molecules are formed, which then form xylose 5-phosphate or 2 molecules of xylose 5-phosphate and 1 molecule of ribose 5-phosphate. This whole pathway has two type of stages, oxidative and non-oxidative. In oxidative stage, the six carbon sugar is converted to 5 carbon sugar, whereas in non-oxidative stage of HMP pathway, the pentose sugar is converted to different type of sugars, and hexose sugar formation occurred. Now see here xylose 5-phosphate and ribose 5-phosphate, one molecule of xylose 5-phosphate and one molecule that is from ribose 5-phosphate enter in the reaction, where the keto group from xylose 5-phosphate, because it is keto sugar and ribose is aldo sugar. So from the keto sugar, keto group is transferred to ribose sugar, which is 5 carbon, and thus pseudoheptulose 7-phosphate is formed. The remaining 3 carbon of xylose 5-phosphate result glyceraldehyde 3-phosphate. This pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate further goes to another reaction that required transaldolase, because this transaldolase cause cleavage of aldol from pseudoheptulose 7-phosphate. The 3 carbon unit transferred from pseudoheptulose 7-phosphate to glyceraldehyde 3-phosphate and then fructose 6-phosphate, or one molecule of fructose 6-phosphate is formed. The remaining 4 carbon unit of pseudoheptulose 7-phosphate then form etherose 4-phosphate. Now come to another molecule, the remaining molecule of xylose 5-phosphate. It then again react with or take part in the reaction of transketolation, where its keto group is transferred to etherose 4-phosphate. So 2 carbon unit from xylose 5-phosphate and 4 carbon sugar, that etherose 4-phosphate, combine and form fructose 6-phosphate. The keto group is transferred to aldose sugar and keto sugar is formed fructose 6-phosphate, and again the glyceraldehyde 3-phosphate is formed. So total summarizing the reaction, 3 molecules of glucose 6-phosphate enter that require NADP, and 3 molecules of carbon dioxide are formed with 2 molecules of glucose 6-phosphate, because the 2 molecules of fructose 6-phosphate which are formed are epimerized to form glucose 6-phosphate, and 1 molecule of glyceraldehyde 3-phosphate is formed, whereas total 6 NADPH are formed with 6 H ion, because these are formed simultaneously. Now first see some differences of HMP shunt or HMP pathway with of that Embden-Meyerhof pathway or glycolysis. This HMP pathway is continue in the specialized tissues only to serve specific function, like in liver, adipose tissue, and here basic requirements are there. They are require NADPH for synthesis of different compounds, and even in erythrocyte cells, testis, ovaries, adrenal cortex, and in lactating mammary gland. They are not required in non-lactating mammary gland. And the cornea of the eye or lens also required the intermediate of the of this HMP pathway are involved in the functioning or basic requirement of the lens or cornea of the eye, whereas it is unimportant for skeletal muscles and also the non-lactating mammary gland, as I told you. When comparing with glycolytic pathway, so this is the multicyclic pathway. We saw that different pathways are involved in the same in the single pathway, and in this pathway, 3 molecules of glucose 6-phosphate enter, producing 3 molecules of carbon dioxide and 2 molecules of 5 carbon residue pentoses, which are then rearranged to give 2 molecules of glucose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. So 2 molecules of glucose 6-phosphate are regenerated and only one oxidized completely. Oxidation is achieved in this process by dehydrogenation, whereas NADP is used as hydrogen acceptor, and whereas in glycolysis NADP are involved. Carbon dioxide molecules is not formed in glycolysis, whereas carbon dioxide is produced in this pathway. The similarities in both pathways, the glycolysis and HMP pathway. So both the pathways require enzymes that are extra mitochondrial or present in the cytosol of the cell, and the pathway is operated or the pathway is continued in the cytosol. Before going to step by step or each and every reaction, first of all, I would like to remind you again that this pathway is basically divided into 2 stages. Stage 1 is oxidative phase, and stage 2 is non-oxidative phase or non-oxidation phase. In oxidative phase, oxidation of glucose is occurred and pentose sugars are formed or pentose phosphate are formed. Whereas in stage 2, pentose phosphate reaction occurs where interconvergence of pentoses occur. And secondly, conversion of pentose phosphate to hexose phosphate occur. Stage 1, oxidation of glucose and the formation of pentose phosphate. That is the oxidative phase. In this stage, first oxidation is occurred. In oxidation, hydrogenation of glucose 6-phosphate to form 6-phosphogluconate is formed, but it is occur via the formation of intermediate, which is unstable, intermediate compound that is unstable, and this compound is 6-phosphogluconolactone, which is unstable and converted to 6-phosphogluconate. The first reaction required glucose 6-phosphate dehydrogenase with NADP, which is NADP dependent, and it then forms NADPH and HI. Then water molecule is added by gluconolactone hydrolase, and 6-phosphogluconate is formed, which is stable. Here you should know how the reaction is regulated, how it is inhibited, and how it is accelerated or activated. The enzyme glucose 6-phosphate dehydrogenase is inhibited by presence of NADPH. When there is excess of NADPH, so this enzyme is inhibited because there is no need to produce NADPH. Whereas this reaction is activated with the presence of enzyme hydrolase or keto or lactonase. Other step of oxidative stage of HMP pathway is the formation of pentoses, and this occurs in 2 steps. First, the 6-phosphogluconate is converted to 3-keto 6-phosphogluconate, and it required the enzyme 6-phosphogluconate dehydrogenase. So again NADP is required to accept HI. So 6-phosphogluconate is converted to 3-keto phosphogluconate in presence of NADP and divalent ion, magnesium, manganese or calcium ion, and NADPH plus H ion are formed. This in the second step, pentose pentose sugar is formed. How? The 3-keto 6-phosphogluconate undergoes decarboxylation to remove the first carbon is removed. So by removal of carbon dioxide, keto pentose is formed. That is ribulose 5-phosphate. The stage 2 of HMP pathway is non-oxidative phase, which involves two types of reaction, in which in the first pentose phosphate reactions in which interconversion of pentoses occur, and in the second hexose phosphate is formed, or fructose 6 phosphate is formed, which is later on converted to glucose 6 phosphate. So in the first pentose phosphate reactions, the ribulose 5 phosphate which is formed is converted to aldose sugar by keto isomerase, which has two further options. Either it converted to D-ribose 1-phosphate, phosphate group is change their position, or it can be phosphorylated in the presence of ATP and magnesium ion. So D-ribose 1,5-diphosphate is formed. And as I told you that ribulose 5-phosphate is first form an intermediate compound, that is anidiol, in which two hydroxyl group are attached to the carbons, which are attached with double bonds, which you can see here. And this anidiol can later converted to D-ribose 5-phosphate. It has possibilities to convert D-ribose 1-phosphate, or it can be converted to D-ribose 1,5-diphosphate by the addition of phosphate group. Now the conversion of D-ribulose 5-phosphate to another pentose, that is xylulose 5-phosphate, another keto sugar, by epimerization. So the enzyme required epimerase, and it converts D-ribose 5-phosphate to D-xylose 5-phosphate. It may be that ribose 5-phosphate epimerized by phosphopentose 2 epimerase to form arabinose 5-phosphate, another 5-carbon sugar. The second step of non-oxidative phase of HMP pathway involves the conversion of pentoses phosphates to fructose 6-phosphate, which then isomerized to form glucose 6-phosphate. So regeneration of glucose 6-phosphate in the pathway occur, and the cycle is continued. In HMP non-oxidative phase, 2 transketolation and 1 transaldolation reactions are occurred. First, see what happened in transketolation and transaldolation. In transketolation, the enzyme required is transketolase, which basically transfer a 2-carbon unit, a ketol group, and it required a coenzyme, that is thymine pyrophosphate. Thus, conversion of keto sugar into an aldose with 2 carbon less, and simultaneously convert an aldose sugar to keto sugar with 2 carbon more, because 2 carbon added and from the substrate 2 carbon is removed. Whereas in transaldolase, the enzyme required for transaldolation is transaldolase, and it transfers 3 carbon moiety, that is dihydroxyacetone, from a ketose to aldose to form another ketose and aldose. In this reaction, basically cleavage of aldol occur, and as it is represented here, 3 carbon containing unit 1, 2 and one here, which is not showing, but it is there, that 3 carbon unit dihydroxyacetone is transferred, and the reaction represented with red line here. So this group, that is dihydroxyacetone, is transferred to glyceraldehyde 3-phosphate. Here see in a elaborative form of transketolation first and then transaldolation. In transketolase, that ketol group is transferred from ketose, that is supposed to be the donor, and this keto group is transferred to aldose acceptor. So another aldose and ketose are formed. Let us see here what happened in this pathway in the transketolation. The xylulose 5-phosphate, which is a keto sugar, the keto group from this molecule is transferred to ribose 5-phosphate, that is aldo sugar. And after taking this 2 carbon containing unit, it results in pseudoheptulose 7-phosphate. And the remaining one is glyceraldehyde 3-phosphate. As I told you, 2 transketolation occur. Another transketolation is occurred when xylulose 5-phosphate, the other molecule of xylulose 5-phosphate, as I told you that 2 molecules of xylulose 5-phosphate are formed. So other molecules of xylulose 5-phosphate, when come in, the other molecule of xylulose phosphate release 2 carbon containing moiety, that is keto sugar, that is keto group, to erythrose 5-phosphate. So the first hexose sugar, fructose 6-phosphate, is formed. So the remaining 3 carbon unit, that is formed glyceraldehyde 3-phosphate. These are that 2 transketolation reaction. Whereas in transaldolation, aldol cleavage occur and 3 carbon unit transferred from pseudoheptulose 7-phosphate, a 7 carbon containing compound, and the 3 carbon units are transferred to, 3 carbon unit is transferred to glyceraldehyde 3-phosphate. So again another fructose 6-phosphate is formed and releasing erythrose 4-phosphate, which we saw that it received the 2 carbon or the keto group from xylulose 5-phosphate. So sequence of this transketolation and transaldolation is here. First, the xylulose, the one molecule of xylulose 5-phosphate, the keto sugar, 5 carbon containing keto sugar, release 2 carbon containing keto group to D-ribose 5-phosphate, that is aldose sugar, in presence of transketolase. So what then form? 7 carbon containing keto, that is pseudoheptulose 7-phosphate, and the remaining 3 carbon unit then form glyceraldehyde 3-phosphate, that is aldose. After that, the transaldolation occurred, in which dihydroxyacetone, the 3 carbon containing unit from pseudoheptulose 7-phosphate, is transferred to glyceraldehyde 3-phosphate. 6 carbon containing sugar, keto hexose, that is fructose 6-phosphate, is formed. So first fructose 6-phosphate is formed, and the remaining 4 carbon unit is then formed erythrose 4-phosphate and a low sugar. Again transketolation occur with remaining xylulose 5-phosphate and 1 and 2 carbon containing unit from xylulose 5-phosphate is transferred to erythrose 4-phosphate. Now it then forms fructose 6-phosphate in presence of transketolase. And the both transketolase enzyme required, as I told you, thymine pyrophosphate and magnesium ion. Simultaneously, glyceraldehyde 3-phosphate and a low sugar is formed. So total 2 fructose 6-phosphate is formed, the ketohexose, and 1 glyceraldehyde 3-phosphate. Saccharose 4-phosphate, which is formed, take part in different pathways. It take part as transketolation in the HMP pathway, as we saw, and it is also participate in the plant and microorganism for synthesis of phenylalanine. The fructose 6-phosphate and glyceraldehyde 3-phosphate, which is formed, is also take part in further reactions. The fructose 6-phosphate epimerized to glucose 6-phosphate as 2 fructose 6-phosphate are formed. So 2 glucose 6-phosphate are formed, whereas the glyceraldehyde 3-phosphate can combine with another glyceraldehyde 3-phosphate and continue the cycle, because glyceraldehyde 3-phosphate with another glyceraldehyde 3-phosphate or diacetone phosphate, which are interconvertible to glyceraldehyde 3-phosphate. So two compound with 3 carbon here can form fructose 1,6-biphosphate first in presence of enzyme aldolase, and then it can further convert it to fructose 6-phosphate by presence of enzyme fructose 1,6-biphosphatase formation. Fructose 6-phosphate molecule formation is possible when dihydroxyacetone is present or glyceraldehyde 3-phosphate group is present. If there is the cycle is continued and if there is glyceraldehyde 3-phosphate present, so another molecule of fructose 6-phosphate is formed. But in the same pathway, in one pathway, one glyceraldehyde 3-phosphate is formed.